Imagine spending days preparing a PCR experiment. Everything is perfect. High-quality DNA, fresh reagents, careful setup, precise protocol, and then nothing. No amplification, no visible band on the gel. So what went wrong? In many cases, the problem is not your PCR machine. It is not your polymerase, and it is not your reagents. The problem often starts much earlier, at the level of primer design. In molecular biology, primers are short sequences of DNA. They define the starting point for DNA amplification in PCR. Without primers, DNA polymerase has no place to begin. In every PCR reaction, we use two primers, a forward primer and a reverse primer. These primers bind to specific regions of the DNA sequence and define the exact segment that will be amplified. Now, imagine your genome as a very large book, a book with billions of letters. Your goal is to find and copy only one specific sentence. If your primers are not designed correctly, they may bind to the wrong location, or they may not bind at all. This leads to failed or non-specific PCR results. That is why primer design is one of the most critical steps in molecular biology experiments. So, how do we design primers correctly? Today we will use a tool called Primer-BLAST. Primer-BLAST is a web-based tool developed by NCBI. It allows you to input a DNA sequence and automatically designs optimized PCR primers. It also checks specificity across the genome to ensure your primers bind only to your target region. Let's walk through an example. We paste our DNA sequence into the input box. Next, we define the region of interest. We specify where the forward primer should bind and where the reverse primer should bind. This ensures that only our target region is amplified. We also define the expected PCR product size. For example, around 400 to 1,000 base pairs. And we set the annealing temperature range, usually between 57 and 63 degrees Celsius, with an optimal temperature around 60 degrees. This helps optimize PCR efficiency. Now we run the tool. Primer-BLAST analyzes the sequence. It searches for optimal primer pairs and evaluates each candidate based on multiple parameters. After a short time, results are displayed. You will see multiple primer pairs. Each pair includes a forward and a reverse primer, along with key parameters, melting temperature, GC content, and self-complementarity. One important factor is self-complementarity. If a primer binds to itself, it can form hairpins or primer dimers. These reduce PCR efficiency and may cause failure. That is why good primer design avoids strong self-binding regions. Another important factor is GC content. An ideal primer has a GC content between 40 and 60%. This ensures stable and specific binding. Melting temperature is also critical. Both primers should have similar melting temperatures, usually within 1 or 2 degrees of each other. This ensures efficient binding during PCR cycles. Finally, we select the best primer pair based on specificity, GC content, melting temperature, and product size. And that is how we design PCR primers using Primer-BLAST. A small step in the workflow, but a critical factor that determines whether your PCR experiment succeeds or fails. In the next video, we will go one step further and learn how to validate primer specificity in detail. If you found this video useful, consider subscribing for more molecular biology and bioinformatics tutorials.